Welcome everybody to our first meeting of 2021. Thank you for coming. I'm really pleased to see such a big turnout so soon after the new year. I guess people are eager to get back to work. We have our usual format of two very brief 15-minute teaser talks. One by Paul Macklin from Indiana University on collaborative modeling work that he's been doing on in-host viral replication and immune response. And then another talk also on a similar subject using different methodologies by Bard Ermentrout from Pitt. I would also really appreciate people's suggestions for future speakers. As a reminder, this meeting is being live streamed. It will be recorded and made available. So I need to let you know that. A reminder who we are. I'm James Glazier, Indiana University, co-lead of the working group with Reinhard Lavenbacher, University of Florida. Web administration by Jim Sluka, and you've had probably emails from Bruce Shapiro, who has been kindly helping organize. You may also have had some emails from John Rice, who has also been helping organize. We do have a Slack channel. So far, it hasn't been widely used, but we would encourage people to use that. And if you need access to it, please ask us and we will provide it. There's also the IMAG MSM wiki page. People who have signed up for the working group are welcome to edit, put material there, and make suggestions. And so we would, again, if you have any technical issues, please contact us with that. Without further ado, our first 15-minute talk will be from Paul Macklin, and I will turn the screen share over to you, Paul, and look forward to having your presentation. Appreciate you being willing to talk to us.
Well, thanks very much, James, for inviting me here today to talk with the group, and wonderful to see a lot of familiar faces and a few new faces here as well. Hopefully this is sharing my presentation, and I'm going to just change it to make sure that you have the full screen view of it. So kind of we have more slides than we should have time for. I didn't realize it was 15 minutes instead of 30 minutes until just I read your email about 2 minutes ago, James. So we will give the super abridged version of these slides here today, but very happy to answer any questions afterwards, one-on-one or in the Slack channel for the group, and particularly be a great place to take the discussion. The work that I'm pointing out here today really is a team effort. There's absolutely no way we could do this by ourselves, and so I want to just thank at the very beginning our wonderful coalition of modelers, immunologists, virologists, and others across the US, Europe, and other places, led in particular by my postdoc, Michael Getz, and PhD student Yafei Wang, who I'd like to put a little plug in, who will probably be finishing his PhD this summer and looking for a postdoc position, and he's fantastic. So thanks for our coalition for contributing to this, and also thanks to sponsors who have helped with this work, both recently the Jane Koskinas to Juliano's Foundation for Health and Policy that gave us kind of an emergency Kickstarter grant, and then more broadly for methodologic funding from the Breast Cancer Research Foundation, the NCI, and the NSF. And so as we all know, COVID is a multi-scale problem. It involves processes going all the way from the subcellular scale all the way to the tissue scale and the system scale, linking processes like virus moving in the extracellular spaces, binding to a receptor, in this case ACE2 receptor, being endocytosed or brought into the cell, releasing and encoding and releasing the virus and releasing its RNA contents, which then eventually is going to start pumping out viral proteins. Those get assembled back. And then get assembled into new virus particles, and then exocytosed back out of the cell and spread in the extracellular spaces and go on to infect other cells. But then, of course, you have a whole bunch of other processes going on. Some of the more important ones we have to bring up here to set up today's talk is that when a cell is infected, it can detect that. Actually, it has its own antivirus responses, in particular interferon signaling, where the cell will start pumping out interferons, both internally to help reduce cell virus replication, and also send that out extracellular as a kind of like an early warning that something's going on here, and cells nearby will then respond to that interferon resignaling by also reducing the viral replication. And then there's a paracrine signaling response where actually a cell receiving an interferon response will send out its own interferon signal to help further spread the word that there's an infection nearby. And of course, then all the other immune processes that you're probably very familiar with, like macrophages scouring the tissue and looking for dead material, and they can become activated. Dendritic cells, again, kind of scouting out the tissue, looking for signs of infection, and then sampling those, and then bringing back what they've learned, leaving the local tissue and going to the system scale, going to the lymph node, where they're going to present what they found, present the antigens, and help ramp up a more specific T cell response. And then those T cells can go back then to the local tissue and start killing off the infection. So there's a lot of multi-scale processes going on here. It's a complex system. We want to use mathematical modeling to help understand this system and to find better treatments. And the work here we're presenting today is really oriented towards COVID-19, but we've written in a very general format that we think is going to be very extensible to a broad variety of viral pathogens. So it's not just COVID-19. So also to get this work done, realizing that this is a very multidisciplinary and complex problem, that nobody has all the expertise in-house, we've been going through a collaborative iterative process. So we're going to kind of work through rapid prototyping. We take a design as we think it is today, we build it and test it as much as we can, and then refine it based upon expertise both from within our group and for external feedback when we get it. Use a multidisciplinary team to do this thing, realizing that this problem is big, so we have to break it up into sub-problems. So we have a bunch of sub-teams that each work on their own part of the problem, fed by a combination of domain experts and coders and mathematicians working together. And all of this coming together is open source software with weekly team meetings to make sure that we're really steering in the right direction. And so progress has been fairly rapid. We kind of view this as a three-phase program, where phase one was kind of the active community building phase, where our group kind of kicked things off and said, let's build a few prototypes and use those prototypes to help kind of look for extra expertise that we know we're missing to come in and start becoming and building the community. Then as that community built up, we started training members of the community how to build model components. And then at that point, we could step back a bit and say that we're now in phase two, where we're not doing all the lifting and all the coding. Now we're a little bit more facilitators, and the community, the domain experts, and the sub-teams then start coming to the fore and driving the effort forward independently on their own individual components. And then our role is more a systems integrator at that point, that we are coordinating the effort across the sub-teams, making sure things are mutually compatible, making sure that everybody knows about everybody else's progress, and helping to debug and facilitate the model implementation wherever we can to kind of keep this thing going. And then we hope to arrive at phase three, which is where we are going to hand this whole thing out to the community and say that this is something that we feel is worth disseminating, and put it out as an open source application, put it on the paper, and then move more to community support and training. So the methodology we're using agent-based models, in particular, a framework developed by my lab called PhysicCell, which is an agent-based, center-based, agent-based model. So here's just an example of a heterogeneous tumor being attacked by little T cells that are going in chemotaxing, testing for contact, latching onto a cell, testing its immunogenicity, and then killing it off based upon the immunogenicity, then releasing and performing a random walk to keep finding more cells. So there's just an example of an agent-based model. More generally in this framework, we're going to be using agents for all the lung epithelial cells, and for, of course, for all the different immune cell types here. And imagine, because it's an agent-based model, it's just an object-oriented programming kind of context. So each cell can have its own ACE2 receptor model and its own trafficking model and state. And each cell can have its own individual viral replication model, which is kind of a nice step of adding some granularity to the model and getting the spatial temporal dynamics of the process. So kind of showing and giving a brief highlight of the iterative progress we've made. We've gone through several versions of our models so far. The first version is a prototype that I actually just developed overnight here on my desktop, starting with a clean slate of code and doing the kind of the bare minimum starter prototype model, where each cell, imagine that you have a kind of a layer of epithelium. Each cell has its own viral replication model, and then you have extracellular diffusion of viral particles. And as a cell gets infected, that is going to be colored more and more yellow here. And the greater the viral load, the greater the probability of cell death. And so we model that as a really basic pharmacodynamic type equation using Hill function. And these black cells are the cells that have died based upon their viral infection, so stochastic event. But you'll see that kind of concentrates near this this ground zero of the infection, and then the infection will spread out, where you see a wave first of increasingly infected cells spreading out the region of infection. So it'll be like a viral plaque. And then behind that, then you'll see a wave of black dead cells that start, emanating out from the dead cells in the center of the infection, emanating outward. And then the white bits of tissue are really where the dead cells have decayed away, which would kind of like be exposing the basement membrane. So a really basic version of model. This took us a few hours to code and put online, and actually put it out as a cloud-hosted app on NanoHub. And then we went out to the community and said, we think we might have something, but we really need help. And so we reached out to colleagues, we put notes out on Twitter, and really live tweeted actually as we were developing this model, and got some help and built a coalition and put out our first preprint within a week. After that, our next version said, well, really, virus doesn't just magically get into the cell. It needs to rely upon endocytosis to ACE2 receptor. And so we added an ACE2 receptor trafficking model on top of every cell. The other thing is we listened to community feedback, said, well, it's really cool to be able to model just one virus particle landing and then spreading infection, but that's not really how it really happens. That the virus particles are landing randomly in the tissue and forming tons and tons of little tiny plaques that then start growing and merging. The other thing we learned is that the virus diffusion coefficient should have been much, much lower. And so we reduced that, again, based upon community feedback, and came up with a second version of the model. So we got much more realistic dynamics now, and we kind of get this idea of how very nonlinear the whole process can be, and this non-trivial geometries of growing and expanding plaques that start growing and degrading the tissue and then merging together. Now, we're missing something kind of big here so far. In particular, there's no immune system. So this is like the worst case of a very extremely immunocompromised patient. So now the next version is really where we started making that transition to community driven. And there's no way we could have done this on our own because we're not immunologists. So we recruited in a lot of mathematicians who are very good at modeling immunologic systems. And a lot of our team members are on this call here today. And then also some wonderful virologists and immunologists. And so we added the first version of immune model where these green cells are macrophages, and they're wandering, kind of randomly patrolling the tissue. And when they find a dead cell that has died, for example, based upon this infection, they will stop there and start phagocytosing that dead material, and then they will activate and secrete a pro-inflammatory factor that's going to recruit in neutrophils and CD8 positive effector T cells that are going to come in and start eating away the tumor. I'm so used to tumor biology, eating away at the infected cells. So here you see some activated macrophages, especially after cell death events. The plaques are still spreading. Let's see here. Now we have some recruited neutrophils that are going in and starting to take up some of the viral particles in the extracellular space and starting to eat dead cells. And then finally, you'll see these red CD8 positive effector cells arriving, and you'll see that they're going to start really attacking the infected cells. Oops. So I'm hoping that I can just pause this, kind of show you the final screen here. You can see that, if you compare the final screen of the model without an immune system, model with an immune system, it has slowed down the spread somewhat. And of course, you can ask all sorts of what if questions, saying, what if you had more T cells or some resident T cells, for example? Or what if T cells did a better job or worse job at killing? And you get different responses as you would expect. So now we're in version 4, and we're about ready to finalize this, working with the coalition here. In particular, now we said, well, one thing we were absolutely missing is a T cell expansion in the lymph nodes. And so we worked really, really hard with the team, also led by Adrienne Jenner and Turundu after over at IUPUI. Here the idea is that now the cells are not just in the immune, just in the local tissue, but they actually will leave the local simulation and pop up in an ODE scale lymph node model. And then based upon what's going on in the lymph node, you can get a T cell expansion, population expansion, and those T cells will come back in then and start attacking things in the tissue. So that was one of the big changes. We also made other changes in the model to, for example, do a little bit better job of a discrete to continuum conversion, that if you have a field of virus particles interacting with a field of a population of ACE2 receptors on the cell surface, you have to be really careful in that discrete conversion so you don't get portions of a virus particle imported into a cell. You want to have discrete events. So we did a little bit of work on that. The other major things, we started adding the interferon response we just mentioned a little bit ago by adding extra ODE models to each individual epithelial cell and adding a few more diffusing fields in the domain. So here's the full-on integrated model that shows these effects together. So now you have more macrophage polarizations. I apologize, I don't have an extra legend here. But now all these different kinds of immune cells are starting to work together much better because they're able to communicate a little bit more. And the interferon response in particular is doing a good job of slowing down the pace of the infection. I do see a question somewhere in the chat window. I'm trying to figure out how to actually pull that up though. Oh, here's a preprint. Thank you very much. And so really, one thing we were very fascinated with was actually the big difference that interferons make in this process. So let's go back and actually turn off interferon signaling. Well, actually, we can turn off different things in the interferon model. Here we said, let's have the infected cells respond to the interferons that they're making, just slow down the replication, but not have that extra paracrine step. And so they'll secrete interferons, but nobody is passing the message along. Oops. So if I click on this, you kind of see, that we have these infected cells popping up, and the immune cells are attacking them just very nicely, but nothing is really slowing down the propagation of that virus. And so at the end of the day, the tissue is still kind of defeated by the infection here. And one particular thing we find is that there's this gap, right? That interferons are really good for slowing down the course of the infection in the initial affected cells, and the immune cells can catch things that have already been affected enough to be recognizable, but there's kind of a gap in between. In particular, cells can pass on their infection to their neighbors, and they can get infected in that gap between when they themselves start slowing down the replication and when the immune cells start recognizing infected things. So the paracrine signaling turns out to be very, very important. So here now we'll turn on just some moderate paracrine secretion that a cell that's not infected will receive the paracrine signal, the interferon signal, and then secrete its own interferons into the environment to kind of pass the message along as an early warning. And now you see that the spread of the infection is just drastically reduced, and there's much better control of that infection. And now ramp it up by a factor of 10, and you can see that, we'll skip through a little bit more, but immune cells are going in, they're working together, they're keeping that infection under control. And you can see a lot fewer infected cells kind of on the periphery here. In fact, if we probably had a better T cell expansion here, T cell response, I'd imagine we'd do an even better job of shutting down this particular infection. Other things we've worked with is particularly with Ashley Ford-Forsyth and her team recently at Oklahoma. I think they've moved now though, of adding some tissue fibrosis underneath this that damaged cells when fibroblasts can come in and actually secrete collagens to add some tissue fibrosis. Because one thing we're seeing now is a lot of people who suffer COVID, they may well recover, but they're going to be suffering long-term effects. And I think understanding that fibrosis and other long-term tissue damage will be critical for understanding the longer term of the disease course. So kind of now other ongoing works, like we mentioned, improving the viral binding dynamics and replication models to better account for this discrete to continuum conversion to make sure we have discrete events. We have a much more detailed model of cell pyroptosis led by some of our colleagues over at St. Andrews. And we're starting to add pharmacokinetics and pharmacodynamics for popular antiviral drugs. Next steps is after we get the version 4 preprint out, which we expect in the next week or so, we're going to start right away on version 5. We need to start adding some negative feedback so that there ought to be anti-inflammatory signals to stabilize the response. In particular, we haven't modeled antibodies yet, so that's another thing. And then one key point is, what about if there's nothing in this model right now that penalizes an overactive immune system? But we know that actually there are bystander effects and that the immune cells can damage cells nearby that aren't infected. And that's really one of the worst cases of a runaway effect. So we're going to start working on that next. Version 6, we hope that by that point that we have most of the major effects that we have, and we'll call this a final release candidate and start fine-tuning the parameters and preparing for HPC-powered analysis and model exploration and start prototyping 3D versions of the model, which should be fairly straightforward to do. And I just want to point out too that this is a really nice example of sharing expertise across domains. So we have biologists, immunologists, mathematicians who are accustomed to working in different domains, coming together and working on a big problem together and trying to make fast progress. And we've learned to see, some of the ways things that do and don't work for managing big teams. And we think that we could transfer these to, these skills and these lessons to other disease processes. And so it'd be really great to see if we can kind of learn and come up with other rapid responses to significant problems. And really a lot of it just comes from the will to work together and a willingness to put together open source models where you can make fast progress and having frameworks that are designed to be able to be adapted quickly to new problems. So I want to just thank you all for listening in to our update. And you can try these models yourself. We actually have an online version of this simulator that you can try out on NanoHub. And the preprint is out there, and we do expect to be updating again very soon with the version 4 model. And I'm happy to break here for any questions.
Thank you, Paul, for a very intriguing and concise talk. We have time for 1 or 2 questions. Unfortunately, our format doesn't allow us for a lot of time, but I hope that people will contact Paul individually for private discussions, or if we don't have time to get all of your questions answered now. Question or 2?
So I have a comment and a question. This is Reinhardt. It's very impressive work, Paul. And I should say there is, as they say, there's an app for that. There's a subgroup for that on crowdsourcing models. So if you're interested in some of the social and technical and platform issues, we encourage you to join our subgroup. As a question, so Paul, have you? I'm sure you have thought about this. Maybe you can comment a bit on what it takes to calibrate and validate models of this type that are connected together by many different models.
So that's a really great question, and we're in some sense approaching this somewhat in reverse, where we want to get to a point where we have a model that has all the right generic components. We know from our immunologists and from our virologists and other members, pharmacologists in our team, what some of the critical behaviors are. For example, the receptor trafficking, endocytosis, release, the inflammatory modeling, the certain immune components. And so first is just coming to a point where you say that you have a framework that is simple enough to be tractable, but complex enough to get the key components we think are necessary for this complex multi-scale system. And once we get to that point where we think all the generic components are working, as we've been going, we've been trying to at least get to order of magnitude correct on most of the parameters, saying, we've been looking at preprints and other data put out by different groups across the world now, which has been quite fortunate, that at least constrain the timescales of different processes. And so, component by component, we're trying to constrain at least the timescales of all the little individual pieces, cell speeds, how quickly they can form adhesions, how long they stick to a cell, how long they interact, all the way up to larger scale things like saying, how quickly do you see interferons at the extracellular level, at the tissue level ramp up? And all of these form constraints on the pieces. We do think that once we have something that's at least in the right ballpark for the general pieces, that's the time we start doing large scale model exploration on the HPC, and then using extra methodologies to try to match more quantitatively to the data as we can get at them. And there are a lot of great techniques to do this, which are now, I think, finally feasible because of high performance computing, particularly approximate Bayesian computation, to help find at least compatible regimes of parameters for the right timescales of behavior. But it really comes down to constraining individual pieces, and then when you put the whole thing together, first to make sure it doesn't fall apart, and then trying to constrain as much as possible. Again, high throughput investigation can also help with that process. But it's not trivial.
I apologize, but I'm afraid we're going to have to move on if we're going to have Bard give his talk.
Okay.
And if there's time after Bard's talk, I'd love to come back and have more questions, because Paul, what you presented brings up all sorts of really interesting things to discuss. And I'm sorry about the format, but I really appreciate that. And I hope that you'll have lots of good follow-on from this. And you're definitely welcome to come back and give another talk when the new version comes out. That would be very great.
Thank you. And perhaps it's a good time to plug your Slack workspace, because that'd be a good place to continue this conversation.